Hello and welcome to the Molecure's R&D Day. Thank you all for joining us today. I'm Katarzyna Mucha and I'm Investor Relations Manager for Molecure. Prior to starting, I would like to share some key details about today's webcast. Please note that certain remarks made by the management during this event may involve forward-looking statements subject to potential risks and uncertainties. Please familiarize yourself with the cautionary statement provided in the presentation, which has been uploaded to Molecure's website for investors section. Please note that today's event is being recorded. The meeting today will be held mostly in English, and there's a simultaneous translation provided. You can opt for either an all English or all Polish stream by using the respective link provided in the invitation. Feel free to type your questions in either Polish or English in the panel. Now, allow me to introduce the presenting team from Molecure. Marcin Szumowski, Molecure's Chief Executive Officer and President of the Board.
Good afternoon.
Zbigniew Zasłona, Chief Scientific Officer and Member of the Board.
Good afternoon.
And Samson Funk, Chief Medical Officer and Member of the Board.
Good afternoon.
Today's event will be graced by two esteemed guests who, as external experts, support Molecure's research, in particular in the mRNA area. Joanna Sztuba-Solińska, PhD, an eminent specialist in the field of RNA recombination mechanisms and investigating RNA structure and function. Currently, she serves as the principal scientist at Pfizer in the Vaccines Research and Development Department. Joanna will be joining us soon. And Jakub Nowak, PhD, an expert in biotechnology and molecular biology, currently doing research developing innovative approaches to characterize RNA stability at the Małopolska Center of Biotechnology at Jagiellonian University and within the Max Planck Research Group. Let me provide you with a brief overview of today's agenda topics. Firstly, Marcin Szumowski will take you through Molecure's development plans for 2024/early 2025. Then Samson Funk will continue with the company's most advanced assets, OATD-01 or OATD-02, both in clinical development. Next, Zbyszek Zasłona will delve deeper into the ubiquitinases program, followed by a focus on Molecure's breakthrough mRNA platform and its future. Then they will have a 5-minute break. Following that, at approximately 2 p.m., Joanna Sztuba-Solińska will step in to elaborate on methods for identification of compounds in developing mRNA modulators, followed by Jakub Nowak, presenting selected advanced methods for studying RNA structure. Today, we place particular emphasis on issues related to the mRNA platform and science behind it. Finally, Marcin Szumowski will return with concluding remarks, and we will have a Q&A session starting more or less at 3 p.m. Altogether, today's event should last approximately 2.5 hours. Okay, now I would like to pass our virtual floor to Marcin Szumowski, Molecure's CEO.
Thank you, Kasia. Good afternoon. We've already been introduced in the top row as the presenters. Two other key employees is the management board member Sławomir Broniarek and Director of Research Operation Barbara Dymek. As Kasia mentioned, we will focus on the research, and a lot of the presentations will be about early stage research, in particular the novel approach of targeting mRNA with small molecules. As a company, we're supported in many aspects of its operations by both the members of our supervisory board and our scientific advisory board. And this is particularly important in the areas where we dig deeper into the science and try to explain mechanisms of action and the approaches of treatment of different incurable diseases, where the role of Luke O'Neill and Bart Lambrecht, two key experts in metabolism and in lung diseases, respiratory diseases, are of great importance. As Zbyszek will mention later, we're preparing publications jointly with these scientific key opinion leaders in their respective fields. Also in the area of business development, we are benefiting from assistance of Paul van der Horst, who's had a long experience in biotech business development with many successes and accomplishments. And in terms of clinical pharmacology and clinical development, we're assisted by Nancy Van Osser, who has worked in a number of large companies and biotechs in Europe. So what's coming up in 2024? The main catalyst events involving our pipeline, which is described here, are going to be taking place both in the clinical development programs as well as in the early stage programs. Obviously, the front runner and the most advanced compound in Molecure's pipeline is OATD01, which is a dual chitinase inhibitor, macrophage modifier, and is already cleared in the United States and the United Kingdom for a phase 2 proof of concept study in sarcoidosis, which Samson will describe in great detail. Our second clinical program, OATD02, is midway through a phase 1 study, and also Samson will discuss the progress and the plans in greater detail. A lot of the presentation will discuss our DuPicuitinase platform, where we have 2 programs targeting DuPicuitinase proteins, which are very relevant in cancer. But the largest bit, not only presented by Zbyszek, but also the experts that Kasia described, will focus on the mRNA, the new approach of targeting mRNA with small molecules to modify the translation of that mRNA. So in terms of key milestones and catalyst events for this year, we're anticipating the first patient being dosed either in the United States or in the United Kingdom, whichever comes first. The target month for that is March. And this should have a snowball effect. Once the first patient is in, the recruitment should accelerate very rapidly because the sites will be initiated, patients will be in the process of screening, and we expect that the recruitment process will take off quite rapidly, especially in the US, where we're working closely with the Foundation for Sarcoidosis Research. The next time point in this study, coming late 2024 or early in 2025, depending on the pace of the recruitment of patients, will be the midterm analysis, which is to take place after 50 patients have been dosed. And this time point will tell us how the study is progressing. I will defer more details to Samson when he talks about that program. But this is an important milestone as well, because it gives us a go-ahead or no decision to be made at that stage. D02, we're anticipating reaching the phase 2 recommended dose or the maximum tolerated dose, or both. And this will allow us to plan forward into expansion studies and continued development in OATD02 in solid tumors and potentially also in acute myeloid leukemia, AML. As most of you may know, we have engaged in developing AI technologies, tools, and methods to accelerate and improve the quality of our drug discovery programs, the molecules that are synthesized and tested by chemists and biologists. And we'll talk a little bit more about that in the later stage of the presentation. Currently, we're focusing these technologies on our early stage pipeline, trying to optimize the compound properties so they have drug-like characteristics that allow these molecules to be developed further in discovery and preclinical development. And obviously, business development is something that drives the company and provides financing for future research and new programs. And the target for this year is signing a revenue-generating collaboration or licensing agreement in one of the clinical programs or the mRNA platform, which doesn't require advancing into clinical stage to be an attractive proposition for partnering and collaborations with pharmaceutical companies. So these are the 5 goals that we have set for this year. And now I'm happy to transfer the microphone to Samson and the pointer so he can describe in more detail our clinical programs.
Thank you, Martin. Let me just give you an update on the O1 study. The program O1 is a phase 2 study in pulmonary sarcoidosis. This is a double-blinded, randomized, placebo-controlled, multi-center study to demonstrate safety and efficacy of OATD-01 in patients with active pulmonary sarcoidosis. You can see on the top the line or the schedule of events we have obtained. This is a study that will take place in the United States, in UK and EU. We have obtained FDA approval, which was an important step for the company in July of last year, and are currently about to initiate the US sites. Then we have obtained MHRA approval, which is the approval in the UK. That was achieved in December, and we are also currently initiating UK sites. So we would expect the first patients to come out of the US or the United Kingdom. In the EU, we are slightly delayed, and we expect the EU approval in end of quarter 2 of this year. As you may know, there is a new clinical trials process in the EU, which obviously is not necessarily fostering the speed of approvals in the European Union. We do expect, though, the first patient to be dosed in quarter 2, or maybe hopefully in March. That's our plan. And one of the important points of the study is to be expected end of quarter 4 this year or early next year. This is the interim analysis. We have discussed with all the authorities that we will do an interim analysis after about half of the patients, which is around 50 patients, have completed 3 months of treatment. This interim analysis is a very important point. It de-risks the study program, and it gives us guidance for the second part of the study. We will conduct this interim analysis on blinded data, but there will be some results coming out of this, although the study has still remained to be blinded, so will not be discussing data of patients, but we can give general guidance on how the further study is to be conducted. And we expect the last patient around in first half of 2025. The study is around 90 to 100 patients, male and female patients with active pulmonary sarcoidosis. It's going to be conducted in about 20 to 30 sites in the US, UK, and in the European Union. And each patient will receive treatment for around 12 weeks. The goal of the treatment is to stop the disease progression and the organ damage, eventually even a reversal of the granuloma in the lung, could be achieved. We will quantify this by conducting a so-called PET-CT scan. This has been rated as one of the most innovative endpoints by the FDA and the Foundation of Sarcoidosis Research. This is the first time this is a primary endpoint. We, of course, will also want to improve the symptoms of the disease, such as the lung function, and we want to prevent further lung dysfunction. And this is measured by typical clinical parameters such as forced vital capacity and also radiographic investigations. And very importantly, also we want to improve the quality of life for patients with that chronic disease. On the next slide, you will see again we have the approval from the US and the UK. Currently, we are on the way to initiate US and UK sites. We will need to resubmit the protocol after introducing some changes in the EU and Norway. And from the grant point of perspective, there are two grants, a PARP grant and an NIH grant currently under revision. We have also, of course, we are not alone. This is has become a very competitive environment. So we are, of course, stepping up so-called branding activities. We have entered into a formal collaboration with the Foundation of Sarcoidosis Research, which is the largest patient association globally, based in the United States. We also participate in upcoming conferences like the WASOC, which is the International Society for Sarcoidosis. There will be a social media campaign together with the official start of the study. There's also a special website in the United States. As you may know, in the United States, it's a little easier to go directly to the patients rather than only stay within the clinics. So that's the O1 current status. Now I'm switching over to our oncology study, which is the program OATD-02. This is the first in human clinical trial. It's an open label single arm study with a dose escalation. We are using a very innovative design, which is called Bayesian design. That's a statistical design which enables you to enroll patients quicker. Although quickness, of course, is a relative measure because you are dependent on the patients and the clinics. This study takes place in Poland in 3 sites, in Warsaw, in Otwock and Bydgoszcz. And we anticipate to enroll around 30 to 40 patients with relapsed refractory advanced or metastatic solid tumors. These solid tumors are mainly colorectal cancer, one of the most frequent tumors in Poland, pancreatic cancer, renal cell cancer, and ovarian cancer. The study duration is approximately 2 years, but this, of course, will always be dependent on when we achieve the clinical and the scientific goals that we are targeting. Now, this is the breakdown of our plans. We do anticipate 6 dose cohorts. We are currently at dose cohort number 3, as you can see, that the 10 milligram cohort. The last patient or the third patient has been dosed end of last week. So if everything goes well, we can complete that cohort in about 4 weeks. After each cohort, we will have a checkup by an independent committee who gives give us a green light to advance to the next cohort. This is a special character of phase 1 studies. You are bound to a lot of rules. As you can understand, this is a first in human study. So patients are first time exposed to a drug. So we need to be very careful. We currently anticipate that we may not have to advance to the 6th dose cohort. However, this will depend on how the patients react, whether they can tolerate the drug, whether we get efficacy, i.e. a tumor response. So this is the plan, but reality can be quicker, but it can also take longer. Depending on the results, we would plan to expand together either alone as single agent or in combination with other agents for further studies in solid tumors and eventually also in hematological malignancies, as Marcin has already told you previously. So this concludes the short wrap-up of our current projects, and I'm transferring you now to the Ubiquity Days project. Thank you.
Thanks, Samson. So now let me update you on what's happening in the preclinical setting, and the most advanced program that we have, that we hope that will enter the clinic soon, is the USP7 inhibitor. This is a pathway which is quite important and mostly described in cancer cells. The pathway of USP7 and NMDM2 and P53 important for the tumor growth and proliferation, but we have explored it specifically in the T cells, and we have optimized our compounds and the screening cascade based on the activation on T cells. So in a way, we found a new way to look at that pathway and a new way to design drugs and utilize inhibition of USP7 to drive T cell activation. And so this is our program, and this is where we are getting all the positive data in cancer agnostic ways. So meaning we have demonstrated efficacy in different models, and what they have in common is that T cell activation pathway, which I have described. This is a reminder of the cellular effect that we observe. It's not easy in nature to find an activator, and I think we did. So we have observed that the USP7 inhibition is inducing interferon gamma in T cells, and at the same time decreases IL-4 and IL-13. When you look at this type of a profile, it kind of shows you an enhanced killing capacity and activation of T cells, and the so-called anti-inflammatory or anti-activation cytokines are down. So that type of balance of that Th1 enhanced over Th2 response is something that you would like to see in patients who fight cancer with their own immune system. Importantly, we have found that results also ex vivo models in situation when we have isolated cells from the cancer in models when we showed efficacy of our compound. And as you can appreciate here, cells which were further stimulated show that enhanced killing capacity. Granzyme B and interferon gamma, both kind of tools that T cells use to kill the cancer, are enhanced when they were isolated from mice treated with our drug. We have shown that mechanism before, but we have since then repeated in different settings, and you can see here that a killing assay. A killing assay meaning we have taken cells from mice which were treated with our compound and mixed them with live cancer cells. And we can observe that when cells were taken from mice treated with our compound, they their capacity to kill is maintained. it would stay for a couple of days in the in vitro system, and in a dose-dependent manner, the killing is enhanced. So this is a functional proof that this cytokine balance towards CH1, which I showed before, is actually doing the job, meaning killing cancer cells. This is a reminder how broad these effects were observed. The USP7 inhibitor has found efficacy in melanoma, breast cancer, lymphoma, and colon cancer models, with nice efficacy in monotherapy. This is also a proof and a nice demonstration using flow cytometry that interferon gamma is enhanced in the cells in vivo by the use of our lead inhibitor, specifically in CD4 and CD8 T cells. That leads to significantly enhanced survival of mice treated with our compound in a monotherapy, as you can see on the left hand on the slide, but importantly in a combination with checkpoint inhibitors. So in that case, PD-1 antibodies. So the increased survival of animals was observed in standalone, but also in a combination. So something that specifically from the clinical perspective is the ultimate readout. What's very important, our observations are also hold true in the human system. In this case, we have worked with human T cells, and you can appreciate from these graphs that the USP7 inhibition would enhance their proliferation capacity and interferon gamma expression as well. So this phenomenon is not restricted to the murine or animal system. It's similar in the human. Another experiment that was done in a CRO, well, it's a co-culture system where we have used two different types of cancer to get mixed with immune cells, specifically peripheral blood mononuclear cells. And we looked at the transcriptomic data coming from these experiments and also the proteome level measured by ELISA. And we observed that in this type of an unbiased study, that granzyme B and interferon gamma were among the most upregulated proteins that found in that system. So that same mechanism holds true in a co-culture system, which is as close as you can get to the real in vivo situation. Importantly, we learned something new in that system, which is the inhibition of VEGF and IL-17A proteins, which are kind of cancer promoting. So when we now look at our program, at this moment, currently, USP7 has not entered clinical development. We follow the competition. There is a couple of companies that work on that target, just as we do. And they are listed here on this slide, like RAPS Therapeutics, Ubiquigen, Pharmatropic, Servier, and so on. They are all in the preclinical setting, and so we are now in the aware of that and trying to get as optimal compound as possible and saving time for the clinical development and work here in the preclinical setting to get all the properties of the drug and a specific indication so when it reaches Samsung department, it will be smooth way. Another program that we have in the ubiquitinase platform is USP721 inhibitor. This is a very nice biology behind it. So we have found in an unbiased study that USP21 is controlling STAT3. STAT3 phosphorylation and activation that leads to the cancer growth and proliferation, migration, and so on. STAT3 is known for years to be an undruggable target. And with our USP21 tools, we have found a way to control it. This is an early stage program. At this moment, we have some data in the cellular system with the early inhibitors, but we are looking carefully at the PD markers. So, for instance, we know that the consequence of USP20 inhibition would be changed mitochondrial translation and metabolism, something that lots of cancer cells depends on, and this is a very elegant finding, how you can have cells that would switch their metabolism by STAT3 activation, and then in a kind of personal way, we can personal medicine way, we can personalize the drug to really affect those cells that switch the metabolism. And so we are planning, and hopefully when we will get the compounds that are good enough to use in vivo, we can try to, in a xenograft model, replicate that data in the in vivo system and show that anti-cancer effect in vivo. And this is visualization of what I talked to you about using the genetic approach. In the USP21 deficient animals, you can see that the STAT3 phosphorylation is inhibited, and this is replicated by the use of our early compounds. At this moment, there is 2 companies that work on USP21. There was a recent publication from the Bayer, and another company that has that program in their pipeline is Mission Therapeutics. And now I would like to focus on the early programs in the mRNA platform, where the objective is to design molecules that bind RNA and prevent its function, specifically translation. This is a research plan that we started over 2 years ago in a collaboration with the International Institute, and we prepared the plan and the pipeline with Professor Janusz Bujnicki, who was collaborating with us on that whole program from the very beginning. So let me go over the pipeline. So initially we have spent some time to identify over 40 mRNA transcripts that would be interesting from the business perspective as targets, and obviously from the biology. There is with this type of new drug design, there is always a risk, the technology risk. So we didn't want to have any, let's say, target validation risk. So the objective in the choice of that mRNA transcript was that this is a target that is validated in different labs and is already in the clinic, or we have a situation when we have an antibody or other biologics as a way at this moment to therapeutically interfere with that specific protein. So we wanted to find small molecules that would target its mRNA, because that's what we do. We make small molecules, and this is also a cheaper way and more affordable way to get a drug that doesn't require hospitalization. Out of 40 genes, we have identified a robust structure, so-called structure, which we also call druggable structure in almost all of these genes. But then more importantly, we wanted to make sure that that structure is functional, meaning that this structure is important for the fate of that mRNA. So if there is specific protein that interacts with it that is important for its translation, we know that we can disturb this type of interaction and have functional consequences. And we have validated them in the wet lab using shape technique, something that Joanna will talk to you about in a couple of minutes. And later on, try to that predicted structure to try to verify it with different techniques. Sometimes with cryo-EM or similar. This is what Kuba will later focus on to prepare us for the virtual screen. Virtual screen that we have done, as I mentioned, in collaboration with Professor Janusz Żubunicki. And after getting the best compounds, we went on to validate them in the lab. The compounds that were designed for that specific structure and predicted to bind to it. We later on wanted to verify it using biophysical techniques. And after that validation in the biophysical assay of the most promising compounds, looking at their binding to specific fragments of mRNA, we have chosen the best compounds and tested them in a cellular assay. And that's actually where we found at the beginning, at the end of the last year, so quite recently, something what we described as a proof of concept, meaning that we have designed in that way that I just described to you small molecules that target mRNA and inhibit the translation, and we have tested these molecules later on to prove that they actually interact with that piece of mRNA in a cellular system. They inhibited the translation of a protein which was encoded by that mRNA. This is examples. These are the examples of the structures visualization, and so there's different techniques that were used, and I'm not going to go into too much of a detail, but that kind of shows you what we defined as the druggable region selection. So it had to be stable, and there has to be some literature, and if there was no literature, we did the job in the lab to make sure that region is functional. We look at the conservation and among different species. That's something that is, from the energy perspective, it's not favorable to keep such a structure. And we look at different RNA isoforms. All that work was done in the first part of the program. And there is, at this moment, there is two ways how we want to get the first molecules in our platform. One is that the way that I described to you, the virtual screen after a 3D structure. The other one is a DEL technique. So DNA encoded library that is specifically then looking for the interaction of the actual compounds with RNA. This is looking at the millions of molecules with a specific and interacting with a specific fragment of mRNA, and then later on, obviously, to follow the same hit to lead and conformation of the activity of these molecules. So we are choosing two different approaches currently. One is that virtual screen that we have done, and the other one in a collaboration with the Wuxi company from China, getting the high throughput screening assay. Once the compounds are active, so the small molecule binding to RNA, we want to verify that binding, and at this moment our method of choice was the SPR, the biophysical testing. We have performed these experiments in Cellvita with our best compounds to further select the most optimal molecules. And to focus now one more time on what we have and why we are so proud and happy with the results of the proof of concept. So this is a slide that demonstrates the shape map data together with the functionality. This is for our actual fragment in mRNA of interest. So after verifying that in vitro, we made sure that it would have the same structure in a cellular system, so in cellular. And then we made sure how conserved this fragment is. At the same time, based on literature search and our work, we made sure that that region is also functional, meaning it interacts with, in that case, different microRNAs which can control its fate. So molecules were designed for that region have chances of interacting either with a protein of microRNA and therefore change the fate of that mRNA and disturb the translation process. The company that we were testing in the virtual screen, the company that helped us with the building of a specific library is a ChemSpace, with which we have a collaboration for quite a while. Internally, we have made a pre-selected library with over 500,000 of compounds. From that library, after virtual screen, we have chosen 376 virtual hits, which when later on bought or synthesized and used in this SPR technique in Celvita, where we got kind of 5 different chemotypes with over 30 hits of an KD lower than 100 micromolar. And after a careful selection, the medicinal chemists have chosen 6 hits to be further tested in a cellular system. So there were certain criteria which were chosen for the compounds, and they are listed here. The compounds from that library had to have drug-like properties. So no aggregation, no redox, optimal log P and molecular weight, as well as PSA. So something that would allow us to test these compounds without any problems in a cellular system. And we also even looked at the safety concern very early at the safety concerns, such as potential to inhibit HERC or CYP. This is data from resynthesized compounds that stopped to be virtual and started to be real life compounds, and there are and used for the cellular tests. And this is the final experiment of where we initially made sure that our protein of interest is present. We made sure that our protein of interest plays functional role, and we looked carefully at the half-life of that protein. This is important because, as you can imagine, if you interfere with the translation, it's very important how long a protein can stay in the cell without new translation. So it really varies. We have proteins that have very short half-life, and they are proteins who are stable for 3 days or more without any new translation being present. So this is all the work that was done in Molecure before we started to test the compound. So having that cell line, having all the knowledge about the protein of interest, we started this experiment, and you can see that in a dose-dependent manner, the cells stopped to lose the translation of that target without any toxicity that was obvious by looking at the housekeeping gene. If we waited longer, we started to see the functional consequences of inhibition of that protein. So that's our very nice data. Yeah, this is what we planned, and this is what we achieved, and we move forward, and I think this is also one of the reasons why we decided to have that R&D day dedicated to the mRNA platform. Is it relevant? What I showed you, right? We didn't show any in vivo data. We didn't show preclinical toxicology, and I talked for so long about it. I think it is important because it has all the potential to be a new way of making drugs. When we start with the structural biology of RNA and discover the world of RNA with small molecules. And this is from the business perspective. This is the reason why it's important. As you can see, the deals in the mRNA field where the new companies, mostly US based and West Coast based, US companies make deals with the big pharma, and all the big pharma wants to have a piece of it. They want to put hands on a new way, new methodology of potentially making drugs that can address pathways or molecular pathways or proteins that are known to be very relevant in a disease, but it has never happened because of either they are intracellular and the antibodies can't do it, or simply the protein is undruggable, and all the research failed to take it out. So as you can see, the deals are made constantly and they are repeated. So once people believe that you have methodology, that you know what you're talking about, the deals are repeated, and that's something that we see as a trend and something that will be happening in the years to come. A collaboration that we have is in our RNA field is very important because a lot of basic research happens here. So as I mentioned, it was a successful collaboration with Professor Janusz Bojnicki, with whom we created this plan, and we plan to collaborate in the future. Joanna, you will hear from her in a couple of minutes. From the University of Freiburg, Michael Wolfinger is helping us with the evolutionary conservation of different RNA regions. And from the University of Michigan, we also know the Chase Weidman, who is helping us with the interactions of proteins with RNA. So all these different aspects that are important. Obviously, we collaborate with Małopolska Centrum Biotechnologii and Max Planck. Not sure about the affiliation.
Max Planck Research Group, yeah.
Max Planck Research Group with Sebastian Glatt and Kuba here, and much more people where we know that we need the expertise specifically in structural RNA biology.
Thank you. Now we'll have the opportunity to listen to our distinguished guests as they delve into the intricacies of mRNA structure, function, and methods to study RNA stability. I'm now passing the conference over to Joanna Sztuba-Solińska, who will be followed by Jakub Nowak.
Good morning, everybody, or good afternoon. Thank you for giving me the opportunity to speak today. I'll provide you a little bit of a background on RNA, targeting RNA, and basically everything that led Molecure efforts towards the new discoveries. So one big question, why do we target RNA? The very important thing to realize is that most drugs that are currently on the market, they target proteins. And unfortunately, there's not that many of those proteins, about 20,000, which accounts to this little sliver of RNA of human genome, which is only 1.5% of it. Within those 20,000 proteins, only about 12%, so that's equal to 3,000 proteins, is disease related. And even though we target these proteins, many of them have problems. There are mutations, there's drug resistance, so all of that kind of limits targets that we can successfully sort of challenge for the disease. Within all of the human genome, there's basically about 70% of coding capacity that is responsible for RNA. So RNA represents this huge field, untapped, undiscovered field that we can potentially use for successful development of new drugs. Next, please. Why RNA? RNA is a key molecule that regulates multiple cellular processes. It regulates transcription of itself and also other RNAs. It can regulate degradation of other RNAs and also cell. But most importantly, it is involved in expression of proteins that oftentimes are dysregulated in different diseases. So instead of targeting proteins that are sort of a product of RNA with the purpose of targeting RNA, we are kind of a step ahead. We are targeting a molecule that is responsible for example, for a faulty protein function. RNA also interacts with chromatin, with DNA itself. It can actually lead to relaxation or more condensation of chromatin, regulating gene expression. It also regulates localization or sometimes dislocalization of other proteins and RNA, and then sensing of metabolites, ions, temperature. So it's truly an amazing molecule that regulates myriad of biological processes. Please, next. But you may ask a question, why RNA is lagging so much behind? There is a historical reasons for it. For a really long time, it was viewed just as a simple messenger, sort of this inert molecule that drives information between a DNA and protein. It took a really long time to get it to this point. In 1947, 1950, this was the view of RNA, simple messenger. 14 years later, scientists were able to isolate messenger RNA for the first time. And since then, discoveries of RNA fields start rolling. We discovered new regulatory RNAs that regulate functions of other molecules. In 1980, there was a very first FDA-approved molecule that actually targets RNA, and those were antibiotics. Here is a picture to the right of different sites on 30S ribosomal RNA subunit that is targeted with different antibiotics that are very well known, including streptomycin or spectromycin. Since then, there was a big breakthrough in RNA field, meaning the development of next generation sequencing technologies. And since that leap into a science, we were able to sequence RNA, discover novel molecules of RNA, coding, non-coding, short, long, and also messenger RNAs. And also, because of next generation sequencing technologies, we are able to gain insight into RNA structure. Next, please. Oftentimes, also, RNA molecule was sort of seen as this very simple molecule. Simple because people thought that it just consists of 4 primary nucleotides, A, U, C, G. That's it. But there is more to the story. Those so-called simple 4 letters of RNA alphabet actually can fold into very complex secondary structures, forming stems, loops, hairpins, bulges, N-way junctions. But as you know, nothing is flat in reality. Everything is 3-dimensional. So even these complex secondary structures can be complexed even more into 3-dimensional space, forming these very complex molecular pockets that have high probability of accommodating and binding different ligands. Another challenge related to RNA is that it is folding and it's very dynamic. So it doesn't exist in one state. It can change its binding partners. So it sort of dances around the molecular space, finding sort of this optimal fold that again can accommodate a ligand. But that sort of activity or that plasticity make it sort of hard to define a fixed structure. And the challenge number 3 is that RNA is highly negatively charged, so it's polyanionic backbone. And as a result, it is difficult to build molecules or small molecules that can attach to not only a structure of RNA, specific structure of RNA, but also this negatively charged backbone. So these 3 challenges initially shape this view that RNA is undruggable target. But then I'll present on the next slide that that's actually not the case. See, on the top, you see this so-called simple structure or primary structure of RNA with the 5' end, the 3' end, and those 4 simple nucleotides. I just mentioned to you that they fold into very complex secondary structure. Helices, stem loops, pseudonodes, it's formed when single-stranded region basically binds somewhere else with another single-stranded region, forming this sort of a knot of long-range tertiary interactions. But besides all of that, there is a three-dimensionality to it, and A, B, C, and D examples represent basically duplexes or junctions that have been successfully targeted with different kinds of small molecules that are currently in clinical trials or already on the market. Next, please. Human messenger RNAs also represent untapped potential when it comes to a very complex secondary structures and that form binding pockets for different kinds of ligands. To the left in green, you see iron responsive element. This is an element that, for example, regulates translation. And right above it, you can see a small molecule that binds to that particular motif in bulge region. So this is a single-stranded region that disrupts a stem, and that particular inhibitor, SNCA, that is found in SNCA messenger RNA, has been shown to be very important for Parkinson's disease. Next in a row is a purple, not next slide, next in a row. It is a purple motif that is a splicing modulator. Right next to this is also another molecule that, or another motif of RNA that binds small nuclear RNA in blue. Those two motifs regulate splicing, and it has been shown that basically improper splicing, exclusion of certain exons can lead to diseases like spinal muscle atrophy. And again, both of these motifs have been found successfully to bind small molecules and regulate the protein expression. In yellow, right next to it, there is an RNA that consists of CUG repeats. These CUG repeats, or in general repeats, oftentimes are natural elements of human messenger RNA, but sometimes the expansion of those repeats becomes toxic, and these elements start to accumulate proteins that basically lead to diseases. And again, in this case, because the structure of that particular expansions are known, it was possible to target it with nafuridine derivatives and basically lift off the toxic properties of these repeated expansions of RNA. And last but not least, to the blue field, you can see another example of atalurin compound that has been shown to induce translational read-through to treat Duchenne muscular dystrophy. So those are a couple of examples of human messenger RNA motifs that has been successfully targeted with small molecules. Next, please. So what do we know at this point about RNA binding pockets? Well, this sort of knowledge is very important to select certain RNA motifs for drug targeting and also to be successful in designing ligands. So we already know that RNA binding pockets have unique properties, as unique as protein binding pockets. They have similar volume, brightness, which is a lack of solvent exposure, and successfully we can target them with small molecules. Except that there are differences between RNA binding pockets and proteins, and it is shown those differences are shown sort of in those pie charts. So majority of small molecule recognition by RNA binding pockets is driven by stacking interactions, which are in blue. There's 34.8%. There are also hydrogen bondings that lead to interactions with the small molecule and the binding pocket of a given RNA. Hydrophobic contacts, which lead majority of interactions when it comes to proteins, here they account for about 18% of interactions. And then ions, magnesium, potassium, they're very important for RNA folding, and also they dictate how RNA interacts with a given molecule. Magnesium and potassium are the most abundant ions in the cell, so no wonder they not only shape the folding of the RNA, but also its interaction with small molecules. Next, please. The very important aspect of binding pockets, RNA binding pockets, is the fact that it needs to have a high information content. High information content means it has to have a structural complexity that makes it unique differentiates this pocket from other RNAs. So whenever we target it with a small molecule, it's going to bind to that particular RNA and only to that particular RNA. And also it has to, but this binding event has to disrupt its biological properties. So here at Molecure, we use a methodology that is called ShapeMap. It's the abbreviation stands for a full mouth name, a selective 2' hydroxyl acylation analyzed by primer extension and mutational profiling. It's a huge word for something that basically allows us to probe RNA structure not only in a test tube, but also it can be done in cellulose, so in living cells, in any biological context that we want. And basically, this way we can resolve the complexity of RNA secondary structure and know exactly how RNA folds and what sort of motifs we want to pick from this RNA large unique space to target it with drugs. Next, please. What about small molecules? What do we know at this point about these small molecules? I showed you a couple of examples of molecules that have been already developed, and based on those, we already know that they have unique properties that actually also reflect immediate properties of RNA binding pockets. So they have a lower octanol water partition coefficient than a typical protein binding compound. This is a measure of lyophility. They have a higher topological polar surface. This is responsible or tells you about the drug absorption. They have a higher hydrogen bond donors and acceptors. I did tell you that the hydrogen interactions also drive interactions of RNA binding pockets with small molecules. They have a higher heteroatom containing aromatic rings. So you'll see that some of the molecules have a lot of these flat rings that differentiate them from other molecules that, let's say, target proteins, and they have, in general, rod-like shape. On the right, you see two sort of databases, public databases that are available that actually list a number of physical chemical properties for RNA binders, and you can basically go ahead and pick from them ligands that can be scanned against your target. These databases are called RBind, Robin, Cerna Bio, and DRTL, but basically they provide untapped resources of molecules that can be initially used as in screens against a target RNA molecule. Next, please. So what sort of strategies do we use when it comes to RNA targeting? I will provide you a very, very general overview of three frameworks. The first framework uses RNA motifs as modules for ligand binding. And it focuses on these modules. It can use them as units in designing selective small molecules. And this sort of approach has been taken by two known labs in the United States. The first one is Hergenrothen lab. This lab actually screens a bunch of molecules against a certain specific RNA motif. For example, the triloop or octaloop. And as a result of this screen, they find specific molecules, and then they can combine them together to bind to a specific RNA motif that, let's say, has an octaloop and then right next to it a triloop. So they use something that is called a fragment-based screening. They basically use these fragments, these small motifs of RNA, and then when they have molecules binding to one motif and another motif, they combine them, and that's how they obtain a selectivity. Disney Lab basically took the same exact approach, but a large scale. So they have a library of these RNA motifs, randomized library, internal loops, hairpin stems, and basically, again, they find a specific small molecules that bind to a hairpin, to a triloop, to a stem, and then they find these motifs within a larger RNA, and they find equivalent small molecules to bind to these small units, and they combine them through a linker. That's how they obtain a specificity of a small molecule. Next, please. Framework 2 is a little bit different. It uses RNA-based chemical space. So RNA molecule here is not used as elements, as units, as I presented before, but it is considered as a whole unique target. So the whole structure, so whole sequence is considered here. And it is believed that RNA is basically this unique target that forms defined binding pockets that can be targeted by specifically designed small molecules. First approach here is a scaffold-based synthesis. So there's a molecular scaffold known to target or to interact with RNA molecules. Here to the right, you have different examples of these scaffolds, aminoglycosides that's basically antibiotics that are known to interact with RNA since 1940. I showed you an example of that. And basically, these scaffolds are used and combined and optimized to target a specific RNA structure. Next, please. Second approach in a framework 2 is RNA biased library. So I did show you the databases that are basically contain small molecules that interact with RNA, and that's exactly what we are using, that sort of information. So we have a focused library of small molecules that are known to bind to RNA structures, and those molecules are used. Basically, they're used to be optimized towards a certain RNA. In here, a very useful aspect is deep learning approaches or AI approaches that allows us to basically mine the information on a given RNA, mine the information on a ligand, and adjust the ligand to its binding pocket. And the next piece, framework 3, is completely different approach. Here, RNA target small molecules are assumed to look like a typical drug targeting protein. So here we are using sort of a traditional medicinal chemistry approaches. There are specific rules that have been coined for targeting proteins, Lipinski rule and Weber's rule, and basically we assume that they're kind of similar for RNA. And we are sort of the focus is taken away from the uniqueness of RNA. It is basically assumed that we can target RNA with similar approaches to a similar degree, just like proteins. And there are examples presented here from riboflavin targets and linezolid that targets 20S ribosome RNA, that these sort of approaches also could be successful for certain types of RNA binding pockets. Next, please. So how do we screen or how do we validate that a certain molecule actually binds to RNA? I'll give you a couple of examples. There are many, many more. I had to sort of narrow down the list just because of the time constraints. But we have fluorescent-based assays where basically we have a motif, RNA motif, we fluorescently use a fluorescent dye that binds to a certain motif. It could be an intercalator, and we basically incubate that motif with a small molecule. Here it's a pink square, and basically that pink square outcompetes fluorescent dye, and that's how fluorescence is released, and that's how we know there is a binding event. Lower part or middle part shows 2-aminopurine assay where basically we incorporate a nucleoside into our RNA motif that has a fluorescent properties. And then when there is a small molecule, which is this pink circle, binding to a given motif, there is a fluorescence release just because the 2-aminopurine changes its properties and it generates fluorescence. And then last but not least, there is a FRAT assay with fragment resonance transfer assay where we label an RNA motif with acceptor, and then we have a small molecule with a donor fluorescent dye, and basically binding of the donor to a certain motif brings it in a proximity to acceptor, and there is excitation and emission changes, and that's how we can generate fluorescence and observe a binding event. Next, please. Another very known assay is a small molecule microarray assay. This one is a high throughput assay where we can basically print a library of small molecules on a functionalized glass surface. These molecules are printed sort of in duplicates, and then we incubate a fluorescently labeled RNA motif with that sort of plate. Then there are a couple of washes to get rid of any unspecific binding, but of course a specific binding or specific interaction of a fluorescent RNA target with a given molecule generates fluorescence. We scan that sort of glass plate, and we can identify which molecule binds to our RNA target. And of course, this sort of assay needs to be validated further. We can validate it by shape map, the method that I was telling you about, where we can basically perform RNA structure analysis in the presence of a given molecule and in the absence of the molecule. And basically, it is believed that a binding of the small molecule to a given motif distorts or affects the RNA structure, and we can see this by probing of basically shape probing. Next, please. DNA encoded compound libraries, that's sort of a novel approach where basically on top you can see there is a small molecule, which is this basically a structure that is basically labeled with a DNA tag. It's a colorful kind of label helix. And then these sort of molecules are attached to beads that again contain that tag. And these beads are next incubated with your RNA targets. And of course, there's a couple rounds of washing to get rid of any unspecific interactions. But basically, a binding of that small molecule attached to the beads with a given RNA targets stays, it generates a fluorescence. And then in order to identify which molecule bound to your RNA, you basically sequence these DNA tags, and this way you can identify what sort of small molecule bound to your RNA target. Next, please. Biophysical assay is something that was mentioned before by Zbyszek. The surface plasmon resonance, SPR, that is a biophysical method that directly measures interactions of the RNA motif with a small molecule. Basically, measure changes in resonance angle or wavelength of a glass surface containing basically mobilized RNA target upon addition of a ligand. And those changes can be recorded, and they can actually provide even more information because you can get affinity, binding dissociation, and other biophysical parameters that are really important for judging whether a given or a small molecule has a potential to be a good drug. Microscale thermophoresis is sort of another biophysical assay where basically you measure the direct migration of a molecule in a gradient of temperature. And of course, that migration changes when there is a binding effect, and you can see it basically in a form of a graph. Next, please. Molecular assays are sort of available in most of the labs. EMSA stands for electrophoretic mobility shift assay, where you basically run a gel that contains an RNA and titrated small molecule. Basically, whenever you see a binding event, there is a shift in the migration of the RNA. RNA-seq, that is what basically drives many of RNA discoveries. It's a next-generation sequencing-based approach where basically after the binding of a small molecule to RNA target, you can see a shift in expression of a given RNA, either increase or decrease. Western blot is another way of measuring an effect of your small molecule binding in RNA, but also on a protein level. And RT-qPCR kind of supports alternative or simpler method to RNA-seq, where you can measure basic lowering of RNA expression upon small molecule binding. Next, please. Phenotypic screening is sort of a last step that has to absolutely follow any RNA drug discovery or any drug discovery, where we need to actually see that a small molecule binding to a given RNA not only affects its structure, not only can affect its, let's say, level of expression, but also it has to have an effect on the biological process that we want to modulate, whether that's alternative splicing, translation. We need to see that. And we need to see that either on a cellular level by using luciferase or some sort of reporter fluorescent assay, but also we have to see that effect on the mice. Otherwise, any discovery of a drug is sort of always on an in vitro or biophysical scale. We have to drive this by a biological model. Next, please. This is sort of my last slide wrapping up my story, but I want to bring your attention to the fact that there are many biotech companies that work on small molecule discovery. Arrakis Therapeutics, Ribometrics, Skyhawk Therapies. every larger biotech company has a team that works on finding small molecules, chemotypes that bind to a certain messenger RNAs, human messenger RNAs that are involved in disease. It is predicted currently that US market that is focusing on targeting RNA molecules will increase to 21% when it comes to spending and basically gaining from that field of study. So this is definitely a hot topic that we should pursue, and there is a lot of promises when it comes to its therapeutic potential. Thank you for your attention.
So, hello everyone. So thank you very much for the invitation to this session. So for the next 20 minutes, I would like to shift the focus a little bit toward more like technological developments that we can do to actually understand and to target the RNA. So basically, as Joanna mentioned, a lot of change in the recent years about the RNA and what it is. So actually, we moved away from the basic concept that this is like just the operating memory that can convey the genetic information into the protein. So at the moment, we know that there's a majority of the genome is actually transcribed as non-coding sequences. But surprisingly, we actually look here from this billions of molecules, we know very little about the structures, and there's the reason for that, because in opposite to proteins, they actually can exist. The RNA molecules they exist in several conformations, and they can change those conformations. They can be locked by interacting with by the interaction with the biological partners, or the changing of the environment, or the modification. So basically what we would like to do is to develop the technique that actually will allow to understand those conformational changes, and this way help guiding targeting the RNA molecules. So we follow the fluorescence way to do that, and for that we developed two ways. We call it in-RNA thermometry and extra-RNA thermometry, depending whether we have a fluorescent probe at a specific position on the RNA molecule, or we're using the external dye. And this allows us actually, as you will see in a couple of examples, study several different aspects of the conformational changes. So for this, it's actually quite versatile platform as well. So we can start from either in vitro transcribing any RNA molecule, we can do chemical synthesis of it, and this fluorescence gives us an access to high throughput screening of the RNA targets, where we're using the biophysical device that is dedicated for very precise measurements of the stability changes on many different formats. And what I mean by that is we can measure in parallel several different molecules. So principle here which we focus is the change of the fluorescence with the temperature. So basically, just to keep it very simple, I don't want to dive into detail, we're focusing on physical-chemical changes that happen when the conformation of the RNA molecule is altering, either via the interaction or by the switching from one conformation to another. And we basically here we're combining the fact that we monitor those fluorescent change with the temperature, with the fact that in the same time the RNA molecule under the temperature regime will unfold. So we basically want to combine those two effects, the stability with monitoring the conformational changes. So here you see basically a simple example of a hairpin, which actually gives us an access that we can use this principle to monitor the changes. So in top you see a free dye, and in the middle is just the strand without any conformation. So you see that when you actually follow those graphs, so in the temperature gradient, you don't see any inflection points. But at the bottom where we have the RNA hairpin, which is labeled in a specific position, we actually can see the transition of the conformation, which is the unfolding of it. So what we wanted to actually do is a little bit more by just checking if we put the labels at a very specific position on those hairpins, we can actually understand certain mechanisms related to the conformation changes. So here what you see is one hairpin that I will use for the following examples, which has the label either at the 5' or the 3' position, and we use the external die, which is this green, that can randomly bind to it. And first, what you see on this graph is that the stability which you observe via the external probe is not changing. But if you actually now look for the stability changes at the specific position of the fluorescent probe on the RNA, you could see that we actually are seeing a different stability patterns. So that gives us the thinking that by just having a specific labeling position can help us understand about the mechanism of the conformational change. So we follow that by basically extending the distance from the loop to the fluorescent probe, and you can see on the right that we actually in this way we can start stabilizing, so we're getting more stabilized readout of the stability. And what we also did is we basically modified the loop sizes, which also affected the stability of the pattern. So by doing that, you could see that we can basically come up with the model of how the hairpin can unfold by just having a label at the different positions. And what is even more, which you probably know from the previous talk, where the RNA has a multiple secondary structures that can interact, we would actually be able to know whether we can do it very locally. So what we did is we used those hairpins where we put them in a label in a 5' and 3' positions, very distincting TM. So this is this orange and blue. And to the left side, you see the stability pattern of the individual hairpins. And on the right side, we basically merge those hairpins. And what is important, you see that the graphs corresponding to the TMs are very similar, which was something which we wanted to actually observe, because that means that we can actually see the changes of the conformation very locally. Of course, this was just showing you an example of how this diRNA thermometry can be used to observe the conformational changes in a very simple hairpin model. But in the reality, this model, the RNA has much more complex structures. So for this, we wanted to validate the diRNA thermometry on the ribose switches, which are quite common genomic elements. The RNA can have the conformation, and there's a lot of different functions that they can regulate. And on the right, you see how many different molecules can actually bind to it. So here we've chosen the SAM2 ribose switch, which is a very interesting and ribose switch that can, upon binding of the SAM molecule, change its conformation from the hairpin to the pseudoknot. And we applied in that case, our method, and you can see it on the right, that we can actually follow the changes of the conformation by detecting an additional inflection point, which is the melting temperature. I don't want to go much into the details. This basically TM1 is the temperature that is specific for the pseudoknot, and TM2 is the temperature which is specific for the hairpin. And we wanted to understand whether we can monitor the changes of the stability of this of the pseudoknot and hairpin upon binding of natural ligands like the magnesium and SAM. As you can see here, that the TM2, which is the graph very to the right, when you add the magnesium, you can actually observe the binding, which is as a readout, there's a stabilization effect. And the graph in the center, this is where when we add the magnesium, we see the stabilization of this specific to the pseudoknot melting temperature, the TM1, which is over the 15 degrees. So this method can be very sensitive for the basically following the binding patterns of the molecules to the RNA. Moreover, we can actually even distinguish the binders from the non-binders using this method. So the SAM and SAH, these are the molecules that are very similar, but the SAH is non-binder for this ribose switch, which cannot actually drive the changing the conformation. And you see here the right graph, the blue, basically there's no change in the stabilization following this method in a concentration dependent following the SAH titration regime. And when we actually do the same with the SAM in the titration regime, we see the increased stabilization of the TM1 from the pseudoknot. And in the last couple of slides, I want to show you another example, which is the tRNA. And this is an interesting platform because the tRNA is known to have the 3D conformation and is also heavily modified RNA molecule. And we use this method to actually explore one of the modifications, which is the pseudoredylation. And on the left side, you see the tRNA with marked position where it is actually in nature modified. And there on the right side, you can see the results from the diRNA thermometry, showing you the effects of the individual modification on the stability of its conformation. And to the very right hand side, you see if you actually have all possible modification patterns on the tRNA, you have the highest stability of this conformation. So we looked also into what could be the mechanism of the stabilization of the cryo-EM, and this is quite amazing because cryo-EM is known to give you information about the structure of large protein complexes, and there's very little structures actually derived by the cryo-EM for the small molecules like the tRNA or the small proteins. And here you can see that basically using the cryo-EM, we can see the shape of the tRNA, which has this kind of an elbow-like pattern, and we can fit very well the map of to the shape of this electron density the model to this electron shape. Sorry, we can fit very well the model to the electrodensity map derived from this tRNA. And then we basically modify this tRNA, and what we observe with the cryo-EM, we can actually follow the changes this small modification have on the tRNA level. So you see here that basically the introduction of the pseudorelation leads to the changes in the elbow region. So where there's this L shape happening, and also we can see the distinct phosphate group in our molecule. We also looked into how this specific modifying the tRNA have an impact on the stability of this L-shaped pattern. So you see that when you actually modify the tRNA and at the bottom, you stabilize the anticodon stem loop, and the modification on this kind of a kink where the elbow shape is happening leads to the compaction of the elbow region. And then we come back to our in-RNA thermometry method, and we wanted to see whether by putting the labels in this core or outside of the tRNA, we can actually see differences at the stability level. And that's exactly what we see here. In the middle, you see that introduction of the labels on the tRNA doesn't change the stability, so it means that we can follow now specifically from the site-specific fluorescence of the tRNA. And to the right, you see basically the stabilization changes happening upon modification. So you have the reference, you have the inactive mutants, and the full modification on the tRNA sequence. And you see that the 5' it is stabilized by the full modification, but this is the core, which is actually the most important for the stabilization from the perspective of the modifications. But the story is not that simple. You could ask why not to replace all the U with this pseudoredylation, as it was done in mRNA vaccines, where they basically use the M1 and the pseudoredylation for the RNA. But you see here on this graph is we did the same thing for our tRNA, and basically to the very right you have this situation where we modified it at a specific position, and you see the stabilization, and these two bar plots in a kind of a center show you where in the tRNA sequence or U's were replaced with the pseudoredylation, and that actually leads to the complete destruction of the conformation of the tRNA. We destabilizing this way. So this is important that for the RNA that has the conformations, those modifications have to happen in a specific position. So here, just to summarize what we are doing, we try to develop the biophysical tools to give you an access for the conformational changes happening at the RNA, and we also following it with using the cryo-EM to understand the mechanism of how this modifications, for instance, can affect the conformation of the RNA molecules. And you would ask why this is important. So I think from the previous talk and you can see that there's much more targets in the RNA domain that we can use to deliver the drugs, and having the proper biophysical tools can actually make it much more easier, like for the molecule, to find out the right molecules to treat the new diseases. So with this, I'd like to acknowledge the team I work with. So very big thanks to Sebastian Glad, who actually couldn't be here with us today. We also collaborate with Janusz Bujnicki that helped us design the models for the tRNAs, and Jean-Ming Su group. And thank you for your attention. And I will probably pass to Marcin.
Actually, I think to Kasia, maybe, as we're now. Oh, sorry.
Yeah, thank you, Jakub. Now I would like to welcome Marcin back for a short conclusion for the presentation with key takeaways, and then we will continue with Q&A.
Okay, so I thought it was the other way around, but I'll be happy to provide the summary now and then move to Q&A. So basically, I think we presented our pipeline starting from the clinical projects to the very early stage drug discovery platform, and there were a lot of presentations about mRNA. But starting from clinical, I think that in terms of next milestones and the news flow, it's the progress of recruitment and the midterm analysis for OATD01 that is going to be an important milestone, which we hope to reach still this year. And it's a de-risking and a value inflection point because it will tell us indirectly whether the compound is safe in patients and whether we can progress and continue the study, which means that it has to be doing something as well in terms of efficacy. We will only know the final results when the study is unblinded and analyzed, but the fact of being able to progress after the midpoint analysis is a very important piece of information, both from the clinical and scientific point of view, but also from the business development point of view. The second asset, the main goal of this study is to confirm the safety of D02 in patients and to establish a safe dose that can also potentially be efficacious in the follow-up studies. So finding the phase 2 recommended dose is a step towards expansions into different indications and into combination therapies, which from the very beginning is the target for OATD02 to guarantee higher efficacy and effectiveness in tumors that are very hard to treat and usually will not respond to immunotherapy. The next area is early stage research and early stage programs. And I think we've heard a very scientific and detailed presentations on different approaches and different technologies and the potential of targeting mRNA with new drugs from our two guest presenters. And I think this is an area where we can explore collaborations. Zbyszek was commenting on a number of transactions, some of them very high level and in terms of financial parameters. And all of these transactions, or the great majority of them, took place at the stages where our company is approaching. So very early stages where you had initial compounds having an effect, modulating the translation of specific mRNAs for a specific disease, and that space is also very promising in terms of business development, both in a format of a collaboration with a larger company. As mentioned, most of the big pharma are looking for making progress in this space as sort of the therapies of the future, and we want to be a part of it, as well as in maybe smaller scale service format where we would work with companies of various sizes who have a gene of interest that is not druggable or is difficult to drug with traditional approaches of targeting proteins. So this is definitely a space that we're very hopeful for, and that can generate not only scientific breakthrough results, but also cash flows if such collaborations or services can be initiated. The last thing that we haven't talked about directly, but that has already been introduced and implemented into our early stage drug discovery programs, whether they're targeting proteins, novel proteins, or mRNA, is the generative and predictive AI tools and models. We have already signed 2 licensees with but mostly with US companies that either have commercial or experimental platforms for AI-aided drug discovery. We're already using these tools and have built up a team of both computational drug discovery experts and medicinal chemists who've been dedicated to this task to use these tools and software to accelerate and optimize our drug discovery process to get to the clinical candidate stage much faster and at lower cost than we have been able to do so before. So I think all these things will have a big impact for the progress of our early stage pipeline in the coming year. And we hope to supplement, the clinical progress and the milestones that we hope to reach in the clinical stage programs with the early stage drug discovery. So I think that that may be sufficient for the summary, and I'm sure there may be questions that will address specific things that the listeners may want to know.
Okay, great.
We can also answer in Polish if the questions have been asked in Polish.
Asked in Polish, sure. We are more than perfect with time, I guess. So we are starting the Q&A session now. Please type in your questions in the panel. We already have a few. So let's kick off the Q&A with the following question. This one is in English regarding the OATD-01. Why do you need to study OATD-01 both in the US and European Union? Is study in Europe necessary for drug approval in the European Union?
It depends. You have to realize that pulmonary sarcoidosis is a rare disease. So in order to get enough patients at the speed that you wish, you will have to expand the study to outside other countries. You also have to take into account the prevalence and incidence rates of the disease in various geographic regions. We know that in African American populations as well as in Scandinavian populations, the prevalence and incidence rates of pulmonary sarcoidosis is about 3 to 4 times higher than in other populations, maybe with some exceptions in Asia. So it's mandatory that you address many sites in different countries in order to get enough of patients within a certain period of time. In addition, your study has to reflect the populations in various countries, in various geographical areas. For example, to give you an example, in the United States, you have to be open within your study to all the genetic subpopulations that are in the US, i.e. you have to have African Americans, Pacific Islanders, Indian populations, Caucasian populations, in your patient population, in your study. Otherwise, it may not be accepted. Obviously, Scandinavian populations you will find in Scandinavia, which is why we have Denmark and Norway as countries that are contributing to our study. Sweden was not available for competitive reasons. They are blocked by another competitor. So this reflects the Scandinavian population in Europe. So that's number one. Number two is that obviously, as you can understand, as a company based in Europe, we should also be conducting the study in Europe. Particularly, there is a relatively high prevalence incidence rate in Poland of pulmonary sarcoidosis in Poland. As a Polish company with a compound invented in Poland, I feel it's pretty mandatory to also do the study in your home country, and of course the neighboring countries.
Maybe one other aspect, Samson, is also that we don't really have a cap for the patients in US, and so if the recruitment starts well and everything is going well, it can be used with no almost limit. But on the flip side, the cost might be higher in the US, right?
Unfortunately, yes, that's correct. The cost of the US is. There was a question actually. If you take Poland as 100, then the relative cost of the US would be 550. So 100 to 550. So you can see the US is very expensive. However, if you look at the average or the mean drug prices for branded drugs in the US, it's about if you take the mean in Europe as 100, then the drug prices in the US is 422. So that somewhat also reflects the price that you can get for a good drug in the United States. That's so the cost and the potential return on investment is quite somewhat in order.
Okay, thank you very much. Now we'll move on with another question. This one is asked in Polish, so I will ask this slowly in Polish so that you can get translation. This one might be for Samson, I guess. Czy wniosek na przeprowadzenie
So the application on the trial on OATD1 was consulted with the Polish Office for Clinical Trials, and the restrictive approach to such trials the first time noticed with regard to your application?
Yeah, this is a little bit of a long story, so I need to make some general explanations. In beginning of 2023, or actually is a new clinical trial regulations in the European Union was put in place. Partially, this was driven by the Brexit, but anyway, it was a new regulation. In all European countries where you want to do the study will report to a so-called reference member state. As the reference member state, we have picked Poland, which is the standard in the EMA process, because as we are a Polish sponsor, it's highly or strongly recommended by EMA to choose Poland as or your home country as the reference member state. So in that case, it was Poland. So Poland speaks also for Poland, but also for the other countries in the EU that we are addressing. Now, with that process, it's a pretty complicated process. There is a process that addresses the authorities and the process that addresses the ethics committees. As you know, ethics committees take care of the ethical conduct of the study and whether the study is harming patients or is in line with the protection of patients. So we have picked Poland as the reference member state. as part of the European putting European legislation into process, in 2023, early 2023, a Polish ordinance was put into place which restricts the radiation exposure to patients. Obviously, as it was a European directive that came into force in 2013, every member state has to have an own regulation per each country. So in Poland, that was again put into place in 2023. And this regulation limits the exposure of patients to radiation. This does concern our study, but also any other clinical study in this country, as well as normal treatment. So if you go to the hospital and need an X-ray because you have an injury, that one will also apply. So there was a certain limit set. In our study, we were convinced by our advice from the Polish Radiation Society, as well as all the KOLs in Poland, that we will be matching the criteria that were set by the Polish regulator. This was also possible because there is new softwares available that are FDA cleared and have a CE mark that would have allowed Polish sites also to do the required investigations. This is 2 PET CT scans within the study to be able to stay below the limit of the what is the legal requirement. For some reason that we don't know, this was still not accepted by the Polish regulator, although all other European countries, including and the US and the UK, have approved this part. So that leads to us being unfortunately forced to not do the study in the Polish sites. If that answers your question, but there was quite a few questions. Is there another question that relates to this?
Yeah, yes, there are a few. There's another one that you have already, I guess, partially answered. I will read the whole question and you will probably answer the second part of it.
Why did Molecure decide that in the procedure for OATT01 you chose Poland as the reporting country? Was it not better to maybe report it in Germany, where the phase 1 trial has been already conducted? Do you think that Orion Centec is responsible for that decision, which is the CRO of the company? Did they make that decision?
The clear answer is no. You cannot blame the CRO. The CRO made us aware of this topic, and also we, of course, were made aware by the Nuclear Medicine Society of Poland by this topic. Nevertheless, we, as I said before, we were absolutely certain or convinced that with the technology that is around and with the software that is actually freely available, that we could have all the Polish sites to to fulfill the criteria set by the Polish legislation. Of note, Poland is the only country that has this restriction. All other EU countries did not have this restriction. The choice for Poland as the reference member state was based on quite intensive discussions that we had with the CRO, but also with the Polish Nuclear Medicine Society and key opinion leaders in the pulmonology and sarcoidosis area in Poland, as well as other European countries, members from the European Society of Lung Diseases, specialists, actually Polish specialists that are in some US sites like Mayo Clinic and so on, and also UK specialists. So everyone was more than convinced that this would not be an issue. And it would have been actually, to be honest, very awkward, that a Polish company with a significant number of Polish sites and Polish patients would not conduct a study in Poland. As I mentioned before, it's standard practice in EMA to go to your home country as your reference member state.
Let me just add one sentence. This was all based on the cost, benefit, and risk analysis. We have assessed the risk of not obtaining the approval, taking into consideration the entire documentation from the clinical centers. The documentation showed that there are methods, technologies to go below the cap, the limit, also the rules of the modified protocols that Polish patients cannot obtain more than 10 millisieverts, which is highly restrictive limit, as Samson mentioned, but there were a number of benefits, large benefits, a large group of interested patients, several different clinical centers with very attractive costs. So we had to make this attempt. This attempt has failed. We do not know the detailed reasons for why it has failed, so therefore we have to go to another country.
Yes, of course, the costs is much a major concern as the CEO of the company. I'm usually not so. I'm more attached to taking care of the patients and the sites, obviously. But if you compare the costs of Poland, as I said before, if Poland is take Poland as 100, then the other countries that we have in Europe, like Greece, is about 120. Germany, France, UK is about 150. Scandinavia is 200. And US is 550 as relative to the cost. So you can understand that Poland is very attractive from that point of view, but also there were a lot of Polish sites and, as Marcin mentioned, a lot of Polish patients. I cannot understand what they've written to us, but there were a lot of patients that were eager to be enrolled in a study like this. And the Polish medical community was also eager to to participate in such an innovative trial.
Okay, thank you. Now another question regarding the OECD 01, asked in Polish, so I'll read this slowly. Czy inne kraje, gdzie spółka ubiega się o zgodę na badanie kliniczne, też zgłosiły jakieś uwagi do dokumentacji i jakich aspektów leku dotyczyły te uwagi?
I didn't get
was there any comments or concerns from other member states about the study and about the drug itself?
We have received quite a few questions. So we had to submit the study in the US first, then in the UK, and in parallel in the European countries. So in overall, I actually have a slide for this. If you want to show the slide, then it's also a breakdown of all the questions, because and the character of the questions that were posed by the different authorities. So it was quite some work for the team to address all these questions. It's the last or the second last slide in the in our presentation. I'm not sure on whether you can. It's in the backup slide section. Good. Next one, please. Next one. Okay. Yeah. Perfect.
This one?
Yes. Thank you. So we started with the US. So you can see on the lower part of the is the US question. So we went for a so-called pre-IND meeting with the FDA, which is a scientific advice, because we knew that it's a very innovative concept, and very innovative concepts always trigger questions from authorities and from ethics committees. So in order to de-risk the program and to have a smooth IND process, we went for a scientific advice with the FDA. So we asked them 4 questions, and we got the answers to those 4 questions and 8 additional recommendations, mainly on the clinical protocol. Further questions with respect to cardiac safety, which were responded by us prior to submitting the IND. Then we submitted shortly thereafter the IND, and the FDA raised only 4 questions, mainly on stopping criteria. For example, if the drug has the tendency to be unsafe, when would you stop the study? What are the criteria for you to stop the study? So they were this kind of questions that we could answer. So as a result, we were approved to proceed with the study. That letter is called Study May Proceed. That's a letter by the FDA. And with that letter, we received another 17 non-hold comments, recommendations, which we, in the meantime, we have also responded to. From the US Ethics Committee, a central IRB, we were approved with no questions, only recommendations to the patient-informed consent. The next step was the UK, the MHRA. We received 15 questions from the regulatory agency, 2 of them non-clinical and tox questions, and 13 for CMC and some for clinical, but mainly it was CMC questions. From the ethics committee, we received 47 questions, 7 towards the protocol and 40 questions with respect to the patient information, so information that we have to provide to patients. Of note, in the UK, there is a special committee which is called ARSAC, the Administration of Radioactive Substances Advisory Committee. This is the most critical one, obviously, because they deal with all the radiation exposures to patients, and they had no questions. They just approved it. Then EU. In the EU, the process is a little more complicated nowadays. Part 1 is you get requests from the regulatory agencies. That includes the Polish regulator. In part 2, you get requests and recommendations from the national ethics committees. So each member state has their own rule, and each member state has their own regulator and their own ethics committee. And then you can see the breakdown. of the requests and the questions we've got. I do not want to go into detail for reasons of time, but you can see that there were 48 questions in the beginning, in the first round, and 6 questions in the second round. Out of those 6 questions, 4 were protocol related, and usually the countries do not disclose, or this is not disclosed on which country has raised which questions. Only in the second round we know that 2 of those questions came from Poland for sure, because they were referring to the Polish legislation. From the ethics committees, we received 80 questions, and you can see the breakdown between France, Germany, and Poland, and so on. But from the ethics committees, we all received an approval, even though also from Poland, with no questions. That's the breakdown of the processes that we had.
Okay, thank you, Samson, for providing clarity on this issue. Another question regarding OATD-01. This one is in English. How many patients do you plan in the US or the UK, and how many in the European Union?
We first of all, we do not have a cap. As you know, we are currently already initiating US sites and UK sites. So in general, what we get as informations from the sites, most of the sites will be able to recruit something between 3 and 6 patients. Which is what they can tell us from the initial patient pool they have reviewed. Obviously, if there's new patients coming in, they will also be checked for eligibility for our study. But again, there is no cap, which means that in case the US or the UK, if they enroll very well, then we would not stop them from enrolling. So first come, first served basis. If there is a good recruiter, we will of course accept new patients coming in. What Spišek is just showing is the incidence rates of pulmonary sarcoidosis patients across the world. Although I have to note that gray does not mean there are no patients. Gray only means there is no data. So for the database. So you can see North America, France, Poland, Scandinavia, UK, Spain, Greece, Turkey, Australia and Switzerland are countries where you would expect many patients to come from. Obviously also in Japan and Korea, but in Japan and Korea we have not taken those countries into account because the regulatory timelines are very, very long. In Japan you need around 9 to 10 months to get your study approved, and in Korea it's similar. It's something between 9 to 12 months, and this is of course too long for us. It was not acceptable, and of course the cost is for bringing those countries in is outrageous for logistical reasons.
Okay, thank you. Another one for OATD-01. Can the endpoints in the OATD-01 study be compared in any way to aTyr Pharma's results?
No, they cannot. We are addressing completely different patient populations and modes of actions. Our study is addressing patients at a very early stage, which means once you are diagnosed with a sarcoidosis, mainly pulmonary sarcoidosis, which is, and you either had corticosteroids, which is the baseline treatment, and are currently in pause, where the treatment is paused because you don't just need it, or if you have an active sarcoidosis but you still do not need corticosteroids. These are the patients that is our population. And the reason for is that from our preclinical data, we believe that we can eventually already revert the granuloma formation at a very early stage in order so that it prevents the disease to progress. 8-year study is a different patient population. In that study, patients are already for quite some time on corticosteroids and maybe also on other more nasty agents. And then you use the 8-year compound in order to reduce the corticosteroid dose, which means that the 8-year compound is given on top of corticosteroids, and their endpoint is to reduce the corticosteroid dosage. As you know, corticosteroids in the long term are associated with significant toxicities and side effects. They are generally hated by patients and clinicians for long-term treatment. So that's a completely different section.
So to what Samson said, I would add that from the scientific perspective, we often say that the sarcoidosis studies would be also a proof of concept for the chitinase as a target in human, and we are the way that the study is designed, that we are comparing a drug. The patients who only take our drug to the control which receives placebo is very informative, because once we have data from these patients, we will be really able to define the mechanistic insights into how our drug work. It wouldn't be possible if we would have a design of a study where we put our drug on top of other drugs. There would be too much noise, impossible to discriminate the mechanistic insights. So that's one other important thing to mention about the study design and all the data that comes from it, which would relate specifically to the inhibition of chitinases.
Yeah, that's absolutely correct. If our let's say our target is to either stop the formation of new granuloma or eventually even reverse the granuloma, so the patients would not get into the damage of the lung. And once patients are on corticosteroids or need corticosteroids for quite some time, for a few months and so, then there is already a lung damage, and damage doesn't really go away. Once there is the damage, that damage will remain and will maybe lead to a scar, which is the hallmark of the disease. But we could eventually prevent this scarring or the damage even to occur. And the double-blinded concept, as well as the placebo, tells us if there is an effect, it can only be our drug, because placebo obviously won't work. So that's the attractiveness of this, and this is the reason why we have a lot of sites, particularly in UK and US, who want to be on the study.
Okay, thank you. Now a question, I guess, for Marcin, also regarding OATD01, but this time, would you share your financial expectations regarding a potential deal in OATD01?
Oczywiście.
Well, if I had the numbers, I would have to announce them in the current report. But we have some points of reference, and I have to use my notes because I don't remember the specific digits. I will find them in a moment, but the last transaction which took place in the area related to pulmonary diseases Well, I will just take it from memory because I have too many notes here. It was around 30 plus million. The value of the entire transaction was around $400 plus million.
Okay, so maybe I'll have another question for Samson in the meantime, and probably Martin, you would just go back to your notes.
Już mam.
Okay, fine.
I've managed to find it. So the recent transaction was from January this year. CS Pharmaceuticals license with Merck. The upfront payment was 76 million, and total due value was 336 million US dollars. And that regarded idiopathic fibrosis of the lungs.
Are there any other indications planned if registration in sarcoidosis fails?
Was the last word fails?
Yes, it was. Yeah.
We do not believe that it will fail. But of course, there are, from the mode of action of the compound, and I'm not sure whether Spišek would like to comment on it. From the mode of action of the compound, of course, there are quite a few diseases where it could work. We believe that sarcoidosis is the best, if you want, is the best way to demonstrate the proof of concept of the drug. But there, of course, there's other diseases, but other diseases would require more patients, longer studies, and a significantly higher investment with an outcome that is, as we believe, not as certain as you could see in sarcoidosis. So that's the reason we picked sarcoidosis as our primary first indication.
Okay, thank you. I think we will move to OATD-02 right now. So the first question is there any interim results publication planned, or we will have to wait till the end of the whole study in OATD-02? And the second, so maybe you can answer this question right now. There's also a second part.
The wording interim results is somewhat misleading. It's a tricky thing. This is an open label study, which means you get results open. you can get results every other day if you want. So there's no. It's very transparent. It's very secret. As you know, this is a dose escalating study. So with every dose that we go, where we can complete that dose cohort, for example, 2.5 milligrams, 5, 10, we're currently at 10. Then we have a so-called safety review committee that looks at the safety data and declares are they fine or they're not so fine. So they will give us advice, which is generally followed, that, for example, that dose cohort is safe. Once that vote is there, then basically that dose is safe and we can proceed to the next dose. So if you want, this is an interim result. Remember, this is not an efficacy study. This is a study targeted for safety. So it's the with every step that we can successfully complete, then we are safer and safer and safer. Of course, if you want, as a side effect, we may see some efficacy or activity, anti-cancer activity. That's usually happens in higher dose cohorts, and that would mean, for example, that if 1 out of 3 or 1 out of 6 patients get a tumor regression, i.e., the tumor shrinks, then this would be pretty open, and this would, of course, be a result that would could be announced to the public, because this is an open label study. There is nothing that should be disclosed, or it can be disclosed. So if you say interim results, yes, we already have interim results. So 2.5 and 5 milligram dosage is safe, so we can use it. We would be allowed to put this dose into studies, but of course we are not satisfied with it. We want more. So the next one will be 10. It's somewhat likely it's going to happen, and then we go further, 20, 25 or so.
Okay, thank you. Now the second part of the question. Are there any specific reasons for delays comparing to original plans? At the start of the trial, 20 milligram dose was planned at the week 25 to 46, and now it's several weeks later.
Yes, we were basing our timing on a very aggressive scenario, which means that we would get immediately a patient in once we open the slot. That actually happens. There are, unfortunately, for the patients, a lot of patients actually waiting in line in the 3 sites. So whenever we open a slot, then we immediately have a patient. But you have to remember, these are very sick patients. Most of them have exhausted all the treatments possible. In colorectal cancer, which is the bulk of our patients, most of the patients have had 5 different regimens of chemotherapy before they hit our study. So, which means, and there's no other drug available anymore that would be available to those patients for treatment. So they are very, very sick. So, as in the natural course, patients may progress during our study. Sometimes patients already go into progression when they are defined. When we enroll a patient, then the patient needs to undergo certain investigations, and these investigations take about 4 weeks for the sites to conduct. And during the 4 weeks, they may progress, and then they cannot be taken into the study. That happened a few times. And this was obviously leading to a delay, because we need to replace those patients. But this is unfortunately clinical reality. Our physician colleagues in the sites obviously always look for patients which are relatively stable, so they may not easily progress, but in the clinical reality, you may still you cannot predict how the tumors eventually will progress or not progress. So, but our physicians at the sites, the investigators, they do their best to have the right patients for the study.
Okay, thank you. Now another question pertaining to OATD02. You have already partially answered, but I think this question needs to be asked so that the audience gets a good understanding of the nature of this study. So this one is asked in Polish, so I'll ask this slowly. W prezentacji za półrocze
In the presentation for first half of 2023 for OATD02, we saw that 20 milligrams is the anticipated target dose. Now we don't see this mentioned. Does it mean that we have not seen the efficiency in patients, or we've seen some toxicity in patients? Or that we haven't seen toxicity in patients, and this dose is going to be moved to a higher level?
The expression target dose is not a good expression. It doesn't actually exist in the world of clinical development, nor in the world of regulators. That's for a so-called dose escalating study in phase 1. There are only 2 types of doses. The one is the so-called maximum tolerated dose, MTD, or the recommended phase 2 dose. That's the dose that you can take to advance to further studies. So maybe the expression target dose came from the modeling of the animals when we saw based on our preclinical data there was modeling performed to see on whether the drug exposure would be sufficient at a certain dose. But this modeling, of course, was not done in humans. This modeling was done on animals. So that's where the expression target dose came from. And when I took over the program as a clinician, I said target dose is not the right expression. So we should not have this because it doesn't actually exist, this expression. So that's the reason we're not using it anymore. It may well be you never know in the first in-human study when you will hit the dose that causes toxicities. Remember, this is a safety study, so we will escalate the doses up to the time point when patients cannot tolerate the doses anymore. This can happen. We are at 10. This can happen at 20. Then we have what you would maybe say the target dose, but it can also happen at 30. It would be it's everyone's interest to get as high as possible. More helps more in oncology, which means everyone is trying to get the highest possible dose. If 20 is tolerable and maybe have an effect, then we would continue to the time point when patients cannot tolerate it anymore. The higher the dose you can finally give to the patient, the higher your chances of drug effect, the higher the chances of efficacy. That's why from the modeling, the anticipation was that we may hit the right dose at 20, but it can also be higher. And again, the modeling was done on the animal models, but of course not on the humans. And humans are sometimes not behaving a little bit differently than mice or rats.
Okay, thank you. Now a question regarding the commercial part. What is the most current competition landscape for arginase targeting drugs? Have it changed in the over the last year? More or less projects are running with this target.
No, it hasn't changed a whole lot. There was relatively few competitors from the very beginning. The programs that are still active or appear active in the pipelines of two companies are Merck and AstraZeneca, who have both preclinical programs. As far as we know, based on the published data and the knowledge of what these compounds look like, neither one of them is a dual arginase inhibitor, and neither one has the ability to penetrate cells and have an activity on the arginase 2, which is in mitochondria or inside the cells. So, and there's no compounds in clinical development. There was one ARG1 inhibitor, which was discontinued, invented by Calithera. So that's the whole competitive landscape that we know of. It's a very difficult target to reach with small molecules, and I think we definitely, by comparison with the competition, have the best drug there is on the market.
Okay, thank you. Now a question regarding mRNA platform. So I guess something for Zbyszek. I'll translate this one so that we can move a little bit faster. Will achieving the milestone concerning proof of concept in vitro in mRNA will allow the company to acquire partners for cooperation by providing services to external companies, And what could be the value of such cooperation? And what does validation of selected fragments of mRNA selected by the external client as a therapeutic target mean?
Po polsku czy po angielsku? Czy wszystko jest?
Whatever works for you.
Znaczy, przede wszystkim to
First of all,
proof of concept.
achieving proof of concept shows that we not only talk about it, but we actually do it.
Jeśli chodzi o
When it comes to
transakcje partneringowe,
partnering transactions, I think that at this stage, they can potentially take place at this stage. And this is something that we are focusing on in our company, so that we prefer the offer, so that we talk with interested parties. We go to Barcelona in March. And before we go, we will be in talks with people with whom we've already stricken contact. We will be reminding them of what we have achieved. This is what a bit of this meeting is about. We presented R&D for such a long time. So the idea was to present the proof of concept and the stage at which we are at. And from a commercial side, as I've already mentioned, there are two different models for cooperation. First of all, what does a therapeutic objective of a foreign partner mean? So it's about us providing a service of assessing the drugability of a target and the unfolding of molecules towards modulation and translation of the specific mRNA. This means a gene which is responsible for producing a specific protein by the translation of a fragment of mRNA, which is not druggable by blocking of a specific protein, the way we do with kinases, when we block the activity of a certain enzyme, and by that blocking of that enzyme function, we limit the amount of protein in the body. So there are certain companies which do not have this know-how, do not have this technology, which we have developed, and they approach us with a specific objective. We assess whether this mRNA serves this function, whether it can be bound to, whether a molecule can bind to it, and whether this binding of a molecule with the mRNA fragment results in reducing the protein. So this is a methodology which is a service. And when it comes to cooperation, and most of the large deals which we presented on the slide are cooperation models, along with some exclusivity for proteins or licenses. This involves larger companies where we use again our tools, our know-how for several targets. We might have our in-house molecules, like this one mRNA fragment, which we are not disclosing, where we have seen that we have proof of concept. So having a few such programs results in it being attractive for a large pharmaceutical company. And there are many companies which are looking for such technologies. So it can be a service, which is a fee for service, along with milestones for reaching specific targets, or it can be a long-term strategic cooperation where we exclusively work with a specific pharmaceutical company in a certain area and transfer rights to a specific pipeline or to specific therapeutic targets. And there, the values you saw, the range which I presented, so it is comparable to transactions in phase 1 trials or advanced preclinical for small molecules.
Another question regarding mRNA platform. When do you expect to select the clinical candidate in mRNA programs?
I think it will still take a bit of time, maybe not this year still.
It's a very difficult question to answer. Obviously, at this moment, when we observe the drug-like properties of drugs targeting RNA, it's not much different than from the standard drugs designed against proteins. However, we the proof of concept is in a cellular system. It's more of a it was defined as an inhibition of translation of the protein, but there has to be much further characterization of these compounds to prove that these are actual drugs. And it is obviously there was a, let's say, preliminary data involved, these drug-like properties, but we are now internally thinking about that program as a full-blown program, and within the couple of the next months, we will do certain experiments, which will then decide how we place it in our pipeline. Once we place it in our pipeline, I think I will be able to answer in a better way in 6 months from now or 5 months from now. When we will decide specifically what indication would it be for and so on. So I'm not ready to answer that question yet, but within 6 months I will.
Of course, there are elements that could streamline this process, such as dedicated libraries that we started to work with for the mRNA target. They differ from the libraries targeting proteins and also AI, because using these libraries, we are going to teach our AI tools how to find those compounds that are likely to bind to specific mRNA sections. So this is going to take several years, probably.
Okay, great, thank you. Let's move to questions regarding the grant decisions. We have 2 of them. Mm-hmm. What's the schedule of the upcoming grant decisions, the FANG grant appeal and new FANG grants, as well as grants from the NIH? Also, there's another question, a short one. Have you already received the money from Smart FANG program, the signing of which we reported at the end of last year?
Let me answer in Polish. Yes, the grant that we have already signed an agreement for, so from the Polish Agency of Enterprise Development for the mRNA platform, the grant from the FANG program we've already received, we've signed agreement, but it still works as reimbursement. We bear the costs and we are refunded. So the 32.5 million is not in our accounts yet. It is going to be to come in as we present the results of our project, but this grant is something that we have already received, the decision, and the agreement is already signed. The remaining grants that we have several in the application process, I think 7 of them, some of the decisions are going to be announced soon. Sooner or later, it's a number of grants. The NIH grant is something that's come in fastest. So these are grants for OATD01 clinical trial in the US, only for the US. And here we've applied for $2.2 million, and another grant is for the amount for the program of the early fibrotic program, and it's some $300,000 worth. It's a preclinical discovery phase grant, mainly concerning in vivo experiments when it comes to costs. So we are expecting these decisions to come by the end of the first quarter this year, and we already know the results of assessments, so they are positive, which does not mean that they guarantee their co-financing, because the NIH has not yet received the budget for this year, so all depends on the availability of funding. But this is something that we are going to learn soon. As far as NIH grant for OATD01, in case we do not receive this funding in this competition, we have a resubmission ready with several comments from the taken aboard that we've managed to rectify some things so far. So we expect to be able to fund, but still we expect to be able to fund the majority of the study in the US, and the EU FENC grants are expected in the second quarter of this year. This assessment is going to be concluded in the second quarter this year. This is a very lengthy process. I will not dwell on that, but there are delays. So I think by the end of the second quarter, we are not going to know the decision for these projects. I think we have three projects for early stages of and also the OATD01. We also plan to submit further applications for the National Center for Research and Development. We think that the National Center for Research and Development can be a bit faster than the FENG program there. So all in all, we've submitted applications for over 200 million zloty and 32.5 million we've already been granted.
Okay, thank you. We have approached the end of our scheduled time, so let's make the following question the final question, and this one regards the overall biotechnology sector and the condition of it. Do you see high interest from institutional investors towards this sector? And is Big Pharma more active regarding partnerings, especially during the conferences, the meetings? What's your overall outlook for biotech for the upcoming quarters?
Okay, so I'll answer this one in English since it's last. So there is definitely signs of improvement. The improvement is related mostly to the decreasing inflation rates in most regions, not all yet. But this is a slow process. So we're still far away from the times of pre-COVID and post-COVID when biotech was flourishing and the IPO scene was bubbling and there were a lot of projects and a lot of investments. Unfortunately, as we went into the higher inflation period, a lot of these investments did not return on investment. So the appetite of investors for high risk projects in the biotech sector really decreased. In terms of the partnering in pharma, there is a lot more activity, but most of this activity there's a lot more M&As compared to licensing agreements, comparing from previous years. And usually these M&As are done at later stages. There are obviously early stage licensing agreements and transactions as well, but the shift is towards more de-risked and later stage programs. So I think what will happen, and hopefully it should happen in the next year or two, there will be more tolerance for risk as the inflation rates decrease, and the sector is sort of rebuilding around success stories and the partnerships that led to development of new drug candidates in advanced clinical stages. And I think there is also growing interest in early discovery, which was not there for the last 1 to 2 years. So this is where we're hoping for the mRNA platform to find traction. So it's improving. There is progress. I think the outlook for the future is positive, but companies are still a bit hesitant to take a higher risk at a lower price rather than taking a lower risk at a higher price, which means later on in the development process.
Okay, thank you. That was our last question. A big thank you to all the presenters today for providing a thorough and insightful update, and to our audience for very active participation. For the complete presentation slides, please visit the company website, specifically the Information for Investors section. Additionally, recordings of today's event will be accessible on Molecure's YouTube channel within the next few days. We appreciate your interest in Molecure, and we look forward to providing you with updates in the weeks ahead. If you have any questions, please don't hesitate to contact the IR team. With that, we hope you enjoy the remainder of your day. Thank you.
Dziękujemy bardzo. Thank you.
Dziękujemy.
Thank you.